All right, good morning, everybody, or good afternoon, good night, depending on where you live. Time differences, I guess. Anyway, right now we're seeing BNGO soar up, and part of this could be due to news on NASDAQ compliance. Part of it could also be due to this news that we saw here with Bionano Genomics highlighting their first 3 days of this 5-day symposium. And also, another thing I wanted to point out was this resistance level at sorry about that. Was this resistance level at $5.93. So if we look at the 10-day chart, we can see that it's hit this level a ton of times. So we saw it hit there, we saw it hit here, we saw it hit here, we saw it hit here, here, and now they broke through it. They're heading to the next one, which is going to be at around 655. So you're going to have to wait until it hits that 655 mark and see if it's going to go up or down from there, at least in my opinion. I think that it's going to be trading within this range between this 593 and 655 mark. Of course, I could be wrong. It could soar up past that. Really depends. But just as a disclaimer before we get started, I just want to say that I don't have a position in the stock, as I've said in all of my live streams and also in all of the videos that I've used covering the stock. So if we zoom in, we can actually see how close it is to that 655 mark. Currently, they're about 8 cents off, so they might be hitting it pretty dang quick. Yeah, so they're trying to hit that 650 high, and then if they break that, they go to 655, and there's enough volume, which there seems to be a ton of this morning, they could soar up past that. All right, I'm going to pull up the symposium right now, and I'm going to mute my mic for a second. All right, there seems to be a problem with my audio output capture, so just give me a second. I know they're playing their elevator music right now, so I'll try to fix that as soon as I can.
So as I've just mentioned, our session today on Thursday, January 14th is focused on repeat contraction, repeat expansion disorders, and other complex molecular genetic disorders. So when we started, we thought that we would have a bit of a theme. So we started, as you saw, on day 1 to day 4, where we are circling back to constitutional cytogenetic disorders. But I want to point out that we have speakers throughout the week from United States, Europe, China, and Australia. Just another recap from day 1, that how are we here today from conventional cytogenetics to next generation cytogenomics. And if you look at the timeline and the evolution of how we are here today, we have seen the evolution of karyotyping, FISH, microarray, sequencing technologies, exome sequencing and genome sequencing, and a number of medical associations who have recommended standards and guidelines for using various technologies for the detection and diagnosis of genetic disorders. And now we are here with a revolutionary technology, optical genome mapping, to showcase its potential in both constitutional genetic disorders and neoplastic genetic disorders. So if we look at where are we in constitutional genetic disorders in terms of diagnostic yield? Taken together, all of the evolution of technologies that I showed on the previous slide, we are still at approximately 50% of a diagnostic yield. What this means is that we have approximately 50% of individuals with constitutional genetic disorders that do not have an answer. And our assumption is that most of those variants and answers are hidden in the genome, where we do not have the appropriate tools, or we have not had the appropriate tools yet to get to a diagnosis. Just to reiterate what I said on day 1, rare diseases are not that rare for the family. And according to the World Health Organization statistics, they are approximately 5,000 to 8,000 rare genetic disorders, which results into 1 in 15 individuals affected globally. Worldwide, there are approximately 400 million people affected with a rare genetic disorder, and approximately 30 million of those are from Europe, and the same number are from the United States. Even if we look at 30 million individual statistics, that equates to 1 in 10 Americans affected with a rare genetic disorder. As I have just mentioned previously, we have American College of Medical Genetics, American Academy of Pediatrics, American Academy of Neurology, and other medical associations from around the world that recommend the detection of structural variations as the first-line test for diagnostic testing of prenatal and postnatal genetic disorders. Structural variants can be among the hardest to detect genomic variants. We have sequencing technologies and other orthogonal methods that allow for the accurate detection of single nucleotide variants at base pair resolution. And we have conventional methodologies that have evolved through past decades that allow for the accurate identification of full chromosomal abnormalities such as aneuploidies. However, we have this range of structural variants ranging from hundreds of base pair to millions of base pair that are dependent on the limitation of a number of technologies to identify these different structural variations accurately. This slide is a courtesy from one of our collaborators, Dr. Eddie Maher from the United Kingdom, where this actually depicts that in a conventional cytogenetic lab workflow, there are a number of technologies that are used either in tandem or in sequence to get to a genetic diagnosis. depending upon the starting material or the specimen and which genetic disorder it is, it can be a very cumbersome process and would require the integration of karyotyping, chromosomal microarray, and FISH. We focused on hematological malignancies and solid tumors in the past 2 days, where depending upon the subtype of leukemias, lymphomas, and solid tumors, it can require additional testing, sequential testing, to get to an accurate diagnosis. On the right-hand side, you see a simple, streamlined Bionano workflow where DNA extraction protocols are available, and from starting from sample to report, it is a very simple workflow. Sequencing technologies have been reported to perform poorly for the detection of structural variants. If we look at short-read sequencing, they have been established to show poor performance for large structural variant detection. On the other hand, long-read sequencing technologies have inconsistent throughput and are cost prohibitive for their application as a whole genome clinical assay. Bionano's optical genome mapping technology has the throughput of approximately 96 constitutional samples per week, has the appropriate cost per sample, and can also detect as low as 5% allelic fraction at 400x step and as low as 1% allelic fraction at 1600x step. Let me introduce optical genome mapping technology with Sapphire. On the left-hand side, you see this beautiful video of these long molecules of DNA that are shooting through these nanochannels on our Sapphire chip, which ensure that single molecules of DNA that are linearized in this process are moving through these nanochannels to be imaged on our Sapphire instrument. In the bottom panel is actually an image from the instrument where you see these long molecules of DNA that are counterstained in blue with these small green dots, which are specific 6 nucleotide sequence motifs labeled that are indicative of these green dots, which show this pattern, which will be used for structural variation detection. As I have just alluded to, optical genome mapping with Sapphire has a simple and single workflow where as little as 650 microliters of blood or 1.5 million cultured cells can be used as the starting material. We have unique and specific protocols to isolate ultra-long high molecular weight DNA, and we use a special enzyme to apply these 6 nucleotide specific labels onto the entire genome. These labeled DNA in solution are then transferred onto our Sapphire chip, where by virtue of electrophoresis, these molecules are pushed through those nanochannels and imaged and then analyzed. We have the high throughput, high resolution imaging of these ultra-long DNA molecules, and these molecules are then aligned to a reference genome and to identify these structural variants. This is an example of how the structural variants are detected, essentially by comparing the patterns of the labels to a reference. In the green, we see our reference pattern, and in the blue, we see the structural variant maps in a given clinical sample. A deletion would result from either loss of labels or the decrease in distance between 2 labels. Similarly, an insertion may result from an increase in distance between 2 labels or additional labels that may be between 2 labels. Copy number changes such as repeat array expansions can be very easily detected based on the label pattern and then the distance to accurately quantify them. And duplications, just based on the label pattern, can determine if their orientation is tandem or if they are in reverse. One of the most important structural variants, such as balanced translocations involving 2 or more chromosomes, can also be very easily identified from the structural variant maps to show the pattern recognition aligning to 2 or more chromosomes. Inversions, just based on the label pattern, can also be easily detected. Along with our structural variation maps, we also have a copy number variation algorithm, which is similar to the log-to ratio seen in an array profile, where you can appreciate the whole genome view from chromosomes 1 through 22, X and Y, where we will be able to easily identify duplications and deletions of a given region of the genome. This is a table showing the different structural variation classes, which I've been showing on each of the days as part of the introduction, where aneuploidies such as monosomy and trisomies can be detected, copy number variations such as deletions, whether they are interstitial or terminal, or duplications, which may be tandem or inverted or inserted into another part of the genome, can be very easily identified. Other complex classes of structural variations, such as insertions, whether they are of known or unknown sequence, translocations, whether they are balanced, unbalanced, or cryptic, or inversions and ring chromosomes, can also easily detected, as you can see from this table. Today, we will be focusing on a number of talks where we will see repeat contraction and repeat expansion disorders and how optical genome mapping has been used. And you can see a few examples here, and our speakers will go into detail about these disorders. We are not a sequencing company, so we do not detect single nucleotide variants. I want to orient all of the viewers and the audience to what they will be seeing from the talks of all our speakers today. All the structural variants by optical genome mapping are presented in the circus plot, where you can see the ideogram of the entire genome, chromosomes 1 through 22, X and Y as the outermost ring of the circus plot. The next ring actually shows the different classes of structural variants represented as different colored dots. And on the right-hand side, you can see that those indicate whether there is an insertion, deletion, inversion, duplication, or translocation. The third ring from the outside shows the copy number variation profile, where any deletion or duplication can also be visualized. And the innermost ring depicts different translocation events or other complex structural variant events occurring. And in this example, we have a translocation involving chromosome 11 and chromosome 22. Now, this data can also be filtered against our population control database to identify unique variants and to filter out benign variants. Our users and customers have the flexibility and ability to filter against a specific gene list or different BET files depending on their application of interest. So please remember this as you see all of the exceptional data being presented by our speakers today. We have a fabulous speaker lineup today, as we've had in the past 3 days. Please remember, we will have a panel discussion and live Q&A after the session, so please stay till the end. We will try to answer as many questions as possible. If we don't get to your questions, we will certainly respond via email. Our first speaker is Dr. Birgit Schule from Stanford School of Medicine, where we will be seeing optical mapping and long-read sequencing data to decipher structural the composition of 80xn10 repeat expansions. This is followed by Dr. Alexander Heushen from Radboud University, where we will see his work on the identification of hidden structural variations with optical genome mapping. Two of our speakers, Dr. Crystal Depien and Dr. Mark Corbett, both will be showcasing work on familial adult myoclonic epilepsy. And then we will have Dr. Viola Alisi from Bambino Gesù Children's Hospital with the work on optical genome mapping for the study of repeat expansion disorders and complex cases. We then have two speakers who will be showcasing the work on facioscapulohumeral muscular dystrophy, Dr. Aaron Bosler from the University of Iowa, and then Dr. Jeroen Depreux from UZ Leuven, who will also showcase the work on Fshd disorder. So please stay till the end, and I look forward to seeing you all in the live Q&A. Thank you.
Thanks so much for the opportunity to present at the Bionano Symposium in this virtual format. I'm excited to share with you our clinical genetic study of a Mexican kindred with an ataxin 10 repeat expansion and tell you more about how we use optical mapping and no-amp targeted sequencing, which supports our understanding of clinical molecular genetics of repeat expansion disorders. This slide gives you some background about the clinical presentation of spinocerebellar ataxia type 10. It's a rare autosomal dominant disorder, presenting first with gait problems, followed by disturbance of speech and eye movements, and some patients develop dementia and mood abnormalities. In addition, seizures are a common feature and usually occur several years after the onset of symptoms. Average age at onset is 34 years, and average disease duration is about 13 years. Only symptomatic treatment like anti-seizure therapy is available to date. Interestingly, SCA10 cases have been primarily found in Latin American countries and are basically non-existent outside of Latin America, except for single cases that were reported in the US, China, and Japan. It's hypothesized that a founder mutation likely arose in an ancestral Native American population when migrated from East Central Asia across the Bering Bridge to North America, and then spread throughout the Americas. The map shows you the prevalence for autosomal dominant SCAs in North and South America, and the percentage of SCA10 of the total SCA population, which is between 5 and 45%, and SCA10 is basically absent in the US and Canada. The pathological hallmark of SCAs are GABAergic Purkinje cell loss and cerebellar atrophy. This is illustrated in this MRI, and you see that circled in yellow. And on the right, you see the neuropathological features from a post-mortem brain from a SCA10 case, and which is predominantly Purkinje cell loss and a reduction in volume of the molecular cell layer. The Kalbindin staining confirms the Purkinje cell loss, and the remaining Purkinje cells are dysmorphic with marked reduced dendrites in the molecular layer. And you can see that in the panel E, and it's indicated by the yellow arrow. So now the genetics of SCA10 is based on an expansion of an intronic pentanucleotide repeat in the ataxin 10 gene, and was first described by Dr. Ashizawa's group in 2000. The normal repeat length is 10 to 32 repeats, whereas intermediate repeats are 280 to 850 repeats in length. And we see full penetrance of the disease over 850 repeats. That means you need to detect expansions that are between 4.2 and 22 kilobases in length, which can be quite challenging technically. So in this study, I want to tell you about how we use optical mapping through Bionano and PacBio no-amp targeted sequencing to resolve this ATAXN 10 repeat expansion. We wanted to analyze DNA samples from affected and unaffected individuals from a SCA10 family and apply no-amp targeted sequencing and optical mapping from Bionano Genomics. Clinically, we wanted to better understand phenotype-genotype correlations. For example, how does repeat length affect the clinical phenotype? How does the repeat composition affect the clinical phenotype? And can we assess somatic mosaicism of the repeat expansion? So as you know, Southern blot analysis can only roughly estimate repeat length and size of a DNA fragment containing these repeat expansions. And while more recently developed repeat prime PCR can help us with the repeat composition, it really cannot resolve the complete repeat expansion, in this case of ATAXN 10, and only reads about 1 to 1.5 kilobases at the 5' end. And as you just learned, we need to sequence through several kilobases, up to 22 kilobases. So the exact size and repeat composition cannot be detected with current diagnostic methods, but next generation technologies will allow us, or already allowing us, to solve these challenges. So this is the 3-generation pedigree from our SCA10 family that we have been following for many years. And you see black and red symbols, which indicate affected cases, and the dots indicate mutation carriers. The proband in 3.8 originally presented in the clinic, in our clinic, with Parkinson's disease about 15 years ago. And we did SCA diagnostic panel for him and also his affected brother, who's actually affected with typical SCA10 and not Parkinson's disease. And we found the ATAXN 10 expansion. But we were puzzled at the time about the clinical presentation of Parkinson's and SCA10 in the same family. So to learn more about this repeat expansion and to understand the underlying clinical differences, we first use high-resolution optical mapping. In brief, this technology relies on fluorescently labeled ultra-high molecular weight DNA, which is then run on a flow cell with nanochannels and imaged. And those images are analyzed and assembled in a genomic map, and we can detect structural variants. So here are two examples. In this case, parent-child, as you can see in the partial pedigree on the right, and in individual 4.3, we found a 6.2 kilobase expanded allele, and in 4.7, which is the daughter, we see a much larger expansion of about 13 kilobases, with a number of smaller expansions indicated by the distribution of the green signal in the histogram below. The distribution of these alleles are also shown in the box plot on the right, which we think is reflecting somatic mosaicism and not technical errors. The second technology we applied was no-amp targeted sequencing, with the goal not only to size the repeat, but also to resolve the repeat composition. On the left, you see the standard workflow for no-amp sequencing by PacBio. And in the middle and right, you have examples on how these data can look like and can be displayed. So to explore the repeat structure, we have waterfall plots. And on the y-axis, you can see the number of reads. And on the x-axis, you can see the length of the repeat expansions in base pairs. To examine the repeat characteristics, histograms are very useful and allow for the confirmation of the allele sizes for the normal and the expanded allele, and can show the extent of mosaicism of the expanded allele. So here are some examples for no-amp targeted sequencing. Since we can sequence through the entire repeat of several kilobases, we can also resolve the genomic composition. And interestingly, we found two different repeat patterns. On the left, in samples 4, 5, and 6, we have a represented in gray, together with ATTCC repeats that are represented in orange. And then samples 3, 4, 4, 7, and 8 show a pure ATTCT expansion indicated in gray. Interestingly, again, sample 4, 7 shows a very large variability of the repeat. And when we do the counting, we can count 66 ATTCT repeats up to 2,780 ATTCT repeats, similar to the data from the optical mapping. We were intrigued by this somatic variability and wanted to see if we can also show this at the cellular level. So it's known that the spliced introns of the ataxin 10 repeat can accumulate in the nuclei and can be visualized using in situ hybridization. Here we used a custom designed RNA scope hybridization probe for the ATTCT repeat. In panels A through C, we depict a positive control for the human mRNA. D to F shows a healthy control, skin fibroblasts without the ataxin 10 repeat expansion as a negative control. And I want to point your attention to panel G through I, which shows skin fibroblasts from the same individual, 4.7 of the pedigree. And that again shows the highly variable repeat expansion. So what we were doing is we were counting all the DAPI cells, and we have 7 cells that show a positive signal that you see in H with the yellow arrow, and there are 4 cells that are completely negative and don't show the RNA foci or the label, which are in red in panel I. So presumably the long expansions lead to prominent aggregates in the cells, whereas the shorter repeats show only small puncta or no aggregation of the repeat RNA at all. So in conclusion, the CRISPR-Cas9 or no-amp smart sequencing technology combined with optical mapping allows for accurate sizing of the repeat expansion, can resolve the entire repeat expansion, and establish somatic mosaicism. Exact repeat sizing and composition is critical for clinical practice and genetic counseling, and really can influence disease penetrance, clinical symptoms, and consequently disease trajectory. Here, I'm at the end of my talk, and I would like to acknowledge my collaborator and, of course, the family who is participating in this research. In particular, there is Javier Jimenez, our neurologist from Guadalajara, and then members of the Chile lab, Jos Palafox, Faria Zafar, and Ani Torres. I'm very grateful for the group at PacBio with Yu Chi and Janet, and then the folks at Bionano, Carl Hong, Joyce Lee, and Jill Lay for helping us generate those data. And last but not least, I'd like to acknowledge Dr. Ashizawa, who is a long-standing collaborator and SCA expert. Thank you so much for watching. And if you have comments or questions, please contact me, follow us on Twitter, or visit our website. Thank you so much.
Hello, everyone. My name is Alex Huyschen, and I'm an associate professor at the Human Genetics Department in Nijmegen, the Netherlands. And it's a great pleasure to talk to you about the identification of hidden structural variants with optical genome mapping. What I would like to share is an experience in using optical genome mapping by Bionano for roughly 20 months by now. Despite the lockdown measures in our own laboratory, we still managed to analyze a total of 250 samples over the last 20 months or so. The huge focus has gone into using optical mapping to compare it to our routine workup for cytogenetics. So that is usually a mix of karyotyping fish and CNV microarrays. And we did that for two applications, leukemia samples and samples with constitutional cytogenetic aberrations. What we have done since then are new studies. For example, we looked at several samples with known repeat expansions. We fine-mapped cases with balanced translocations. And we also have several lines of research where we're interested in very low-level mosaicism for structure variants. And again, optical mapping is a very suitable tool for that. But what the main focus of today's talk will be some of our unsolved rare disease research cases. And I'll guide you through some of that work in the remainder of the talk. Just getting back to the very first 2 points, our clinical utility studies and the proof of concept studies we've done so far, two of my great collaborators are presenting that work in this cytogenomics webinar series. That is Dr. Leila El-Khattabi talking about constitutional aberrations in 85 samples, and my former postdoc and now independent scientist in Oulu, Finland, Dr. Tuomo Mantere, he will present our work on 52 leukemia genomes. So I hope you'll attend those seminars as well, and if you're interested in more underlying data, I would refer you to the two bioRxiv preprints that we already have posted. But now to the main topic of today's seminar. Bionano may really enable us to find novel research findings. And those in particular should be structure variants that so far remained refractory to any detection by previous technologies. Some people also refer to those as being in the camouflaged parts of the human genome. And what kind of rare disease cases are suited for this? Think, for example, about families or cases where you already know the disease gene locus for sometimes quite a long time, but so far sequencing has not succeeded in identifying the disease-causing mutation. Other cases we've included so far are classical recessive disease cases where a mutation was found by sequencing on one of the two alleles, but so far the second mutation remains undiscovered. And there again, speculation may be that the second mutation is a difficult to identify structure variant. What we also focus on are hidden structure variants that occur de novo. We know that the de novo mutation rate for structure variants is seemingly quite low. So if you go for patient-parent trios, we should be able to also identify those systematically using optical mapping. Where do we retrieve our samples and case series from? That is usually directly from several collaborating groups in our very own department, so the Medical Center of the Radboud University, where next to my own group, we have 17 other PI groups that work, at least a lot of them work on rare diseases. Then we are engaged in several European and national consortia that also focus on rare disease research, but we're also open to other collaborative studies, and I'll show you more details about that. Just the high-level summary of all the research projects run so far. Altogether, we have run 20 research projects, and we have extremely interesting leads for at least 5 out of those 20 projects. Those can be projects that consist of just one sample in some occasions, but sometimes a series of up to 10 samples also for other projects. And I thought the best would be to highlight 2 cases where we recently discovered interesting findings. Here's the first case. And this is really illustrative for a larger series of experiments that we usually do. The model disease that we study a lot is severe intellectual disability. And that, especially in the Western and outbred population, is very often a sporadic disease and very often is caused by de novo mutations of all kinds. De novo point mutations, as well as de novo structural variants. And to find those more systematically, we study patient-parent trios a lot. So affected patients and the unaffected parents are run in parallel. And in a small series of samples that I present today, we have done all the previous assays already. So these are patient-parent trios that underwent CNV microarray analysis, exome sequencing, and genome sequencing, and most recently, even long-read genome sequencing. And we used 15x PacBio coverage for that. And unfortunately, none of these previous assays identified a disease-causing or likely disease-causing mutation. And therefore, the very simple and straightforward question is, can optical mapping identify the so far hidden de novo structural variant, and may that variant even be linked to disease and may therefore be a novel candidate variant? So what we've done is the series that I'm describing here are just 5 trios. And actually, we only identified and validated a single de novo structure variant in 1 out of the 5 trios. And I'll just guide you through the filtering process that we underwent. So this is a typical trio, and I show you the structure variants called in the proband, the patient. So we usually find 6,000 to 7,000 structure variants per case. By just very simple filter strategies, for example, by only looking into the private structure variants, hence all the structure variants that don't overlap with structure variants in the 200 controlled genomes available to us, we already reduce the number of structure variants dramatically, usually to something in the range of 30 to 50 events per case. But then, if we use parental samples in addition, we can just click once more and look for all the variants that are not present in mom and dad. And in this case, that was only exactly 1 aberration, 1 deletion that is seemingly de novo. Before I show you details of this 1 deletion, I just wanted to make 2 important points. What do we already learn from this exercise on its own right? This already teaches us that de novo mutations for structure variants that are larger than 500 base pairs in size, that de novo mutation rate is probably relatively low, similarly low to what we expect from array and genome sequencing. It furthermore also shows us that optical mapping has an extremely low false positive rate, because de novo mutations would enrich for false positive findings, or the filter approach of de novo mutations. But by very simple filters, we really just end up with 1 true event and not a single false positive. So that is very encouraging in terms of quality of the optical mapping. So this is the one de novo event that we identified, and it ended to be in one of the most complicated regions of the human genome. This is a region in chromosome 17, Q21. And that is an extreme, here in this region, the human genome shows a large cluster of segmental duplications. And what we identified is that this individual, the patient, carried a paternally inherited duplication of this locus, but in addition, a de novo deletion just exactly downstream of that, most likely affecting and disrupting a gene known as NSF. This deletion was validated simply by designing breakpoint spanning PCR primers. And here you hopefully appreciate that there's a successful band of the mutated allele present in the patient, but not present in mother nor father. This PCR fragment was then sequenced using PEG bioamplicon sequencing, and this validated that this truly was a de novo deletion of 121 kb in size. So a pretty large deletion. And again, it was not identified by any previous technology. What are now the details of this deletion? As I said already, this happens in the 17q21v1 region of the genome, really just affecting the segmental duplications that are downstream of the colon de Vries microdeletion syndrome region. Most likely, this disrupts the gene NSF. However, interpretation of that is not trivial because NSF just upstream has a pseudogene NSFP. What is known about these two genes that it's extremely copy number variable in the general population. So there have been studies that propose that there's between 2 and 8 copies of NSF, or likely the combination of NSF and NSFP. So the big question is, while we now identified the very first de novo structure variant in this case, it is intriguing to speculate whether the haploinsufficiency of NSF, where this event most likely leads to, could potentially be a cause of disease. And that really requires further research. So if anyone is aware of deletions of NSF, I would be very curious to get in contact. There is, however, very intriguing recent work, because point mutations in NSF in a study from Japan have been linked to an epilepsy phenotype. So different phenotype, but also the mutations are different. They are point mutations, and the authors of that manuscript explain that they most likely affect the gene function and the protein function by a dominant negative effect. So still the question would be whether simple haploinsufficiency of NSF could cause a disease, but we may have the first case here, so hopefully we identify additional cases in the near future. I brought a second example today to illustrate what we can identify by in terms of structure variants by optical mapping. And that is a collaboration with an old colleague of mine, Roland Kuiper, who now works in Utrecht, also in the Netherlands, in the Prinses Maxima Center for Pediatric Oncology. And one of Roland's interests is a very rare but aggressive form of cancer that is usually diagnosed in childhood, and that is known as atypical teratoid rhabdoid tumors. Usually those present as brain tumors, but can also affect other parts of the central nervous system. The cause for these tumors to occur lay in a gene known as SMARCB1. In quite a lot of cases, there's somatic mutations in SMARCB1 happening early in development. But in roughly 1/3 of all cases, there's already a predisposing germline mutation in this gene, and a second somatic hit then happens in the cancer itself. So Roland and his clinical colleagues have studied a Dutch family with a strong suspicion of a germline mutation in SMARCB1 already, based on the phenotype identified in one of the children. However, so far, no mutation was identified, despite huge efforts. And the previous efforts included diagnostic Sanger sequencing, MLPA, and whole exome sequencing. And none of that identified a mutation in SMARCB1. In addition, Roland's team also applied whole genome sequencing on research basis, but again, failed to identify the disease-causing mutation. So therefore, Holland sent an EBV cell line to us. We isolated ultra high molecular weight DNA from it and performed optical mapping. Similar to the filtering strategies that I've shown you before, we just look at all the rare variants that we identify in this genome. And here you see a list of events. So it's 12 insertions and 20 deletions. In total, 35 rare structure variants in this genome. And then we obviously already had an extremely good candidate locus. What you see here is an event that was called exactly in the gene SMARCB1. So that's one of the 35 private structural variants in this person's genome. If I zoom into the details, you will see here that there is an insertion of close to 2.8 kilobase pairs that also creates one new label that is not present in the reference, but it is present in the assembly of the patient's molecules that we studied. So this is an insertion that happens between, most likely between the introns 1 and 3 of SMARCB1. And we dared already to speculate on this. Highly likely this is a so-for missing germline mutation that gives a very strong predisposition to ATRT. We did not know the exact sequence of this inserted material. So therefore, Holland's team tried to validate by PCR various forms of PCR and resequencing, but unfortunately that remained unsuccessful. So leading to the speculation that it must be a complex part of the genome inserted in this very spot. So therefore, at the end, we decided to perform PacBio hi-fi sequencing. So we created a library of 20 kilobase pairs and sequenced the genome with an average of 8-fold coverage. And that validated the insertion, but also made it more refined. So thanks to the sequence context we now get from the long-read sequencing, we now know that this is an exactly 2,763 base pair insertion in intron 1 of the SMARCB1 gene. If you look at the sequence context, we now also understand that it is a retrotransposon insertion with quite a repeat structure at either end of the inserted material, and that is the likely explanation why PCR and even genome sequencing by Illumina failed so far to identify this event. I think this is a really tough region to sequence, and the context of it does not make it easy to fish it out and see it. So as such, we now believe that this truly is the validated germline structure variant that gives the strong predisposition to ATRT in this family. However, how exactly an intronic retrotransposon insertion in an intron leads to a loss of function allele is still further studied by work on RNA level by Holland Kuiper's group. But interestingly, you could now ask yourself whether there would be more cases where we have these kind of jumping retrotransposons that may disrupt genes. And seeing the difficulty of identifying this one by other classical means, you may ask yourself, how often does that happen? And how often does it happen in so far unsolved cases, even after exome and genome sequencing? So after highlighting these two cases, I just want to finish with a little summary. From all the data we've generated over the last month, I see a tremendous potential for optical genome mapping to replace classical cytogenetics, and that is in particular karyotyping, CNV microarrays, and FISH. For the research cases I've presented today, I hope I gave you a first flavor that the technology can identify hidden structure variants, and in particular variants that have been missed by so far available NGS approaches. I just tried to highlight that with the two examples, a de novo deletion that affects the gene NSF1 and a retrotransposon insertion in the gene SMARCB1 as a likely pathogenic germline mutation. Very extremely recent research findings include several other findings. So we just recently identified a hidden inverted duplication in a family suffering from genetic forms of blindness. in which the locus was known for more than 20 years, but the disease-causing mutations on DNA level remained unknown. We've also started to see the first de novo structure variants in clinically defined syndromes, and we're following up on those. We also managed to fine map balanced translocations to understand the breakpoints in greater detail and understand whether balanced translocations either disrupt genes or disrupt the regulatory regions of important downstream genes. And for some samples, we now have been able to measure long repeat expansions and repeat expansion disorders that so far by classical means were unmeasurable. To sum it up in simple words, I think optical genome mapping by Bionano offers clinical and research benefits and is already enabling us to really live the dream of a cytogenetics revolution. So that is extremely exciting to us and hopefully to many others. Finally, I'd like to acknowledge everyone involved in the studies I presented today. That is, of course, Roland Kuipers' group from Utrecht, everyone at Bionano, but in particular also Tuomo Mantere and Cornelia Neveling from our team in Nijmegen. And I'd like to thank you for your attention and be happy to take questions. Thank you very much.
So I'd just like to start out my talk by thanking Bionano for their opportunity to share our work with you, and also to say what a thrill it is to sort of share the podium, if you like, with Crystal Depian, who's been a fantastic collaborator for us on this work. And so today I'm talking about these new class of repeat expansion disorders, which are AT-rich pentamers, and specifically in the cause of familial adult myoclonic epilepsy. Just as a brief introduction, I'm from Adelaide, which is in South Australia. And if you have any questions about this talk, please feel free to contact. So today we're really talking about repeat expansion disorders, and there are actually now over 50 Mendelian versions of repeat expansion disorders. And if we think about these disorders in the context of a typical gene model, a coding gene model, these repeats can expand and cause pathology from anywhere within this model. So promoter, 5 or 3' UTR, coding or intronic regions. These repeat expansion disorders are characterized by the changing in the size of these repeats from smaller to larger, and that can occur both somatically and across the germline. And the mechanisms behind this are believed to be tied to DNA repair, replication, or recombination. As a result of this unusual dynamic phenomenon, we see in these families the phenomena of anticipation, where disease onset is typically earlier and symptom severity can sometimes be more severe in successive generations of a family, and this is typically correlated with an increase in the size of the repeat. In terms of how these repeats cause disease, it can depend to some extent on where the repeat occurs. So if it's in the coding region, for example, you can get a novel protein function. But even for the non-coding repeats, you can get altered levels of gene regulation. The repeats can be transcribed, and that RNA can form toxic foci, so the RNA binding proteins can be sequestered in the cell or cause aberrant splicing. And then also the repeats themselves can be translated by a phenomenon known as repeat-associated non-ATG translation to form these smaller peptides corresponding to the repeat sequence, and they can be toxic as well. So the disorder that we're interested in, and I'll tell you, we've been looking at this disorder for a long time, is familial adult myoclonic epilepsy. It's characterized by typically a cortical myoclonus, which manifests as a hand tremor predominantly, and that can get worse with exertion. In at least 30% of cases, many individuals go on to also generate generalized tonic-clonic seizures or myoclonic seizures, basically epilepsy. As I said, it's an adult onset disorder, an autosomal dominant disorder, and the onset of the tremor typically occurs in the second to third decade of life. And the disease is mildly, but not very highly progressive. We know of 6 genes that are linked to FAME, and today I'll mainly be talking about STAR-D7, and I know Crystal will be talking about the MARCH6 gene causing FAME3. And what's peculiar about all of these genes, even though they're completely functionally unrelated, is that they all have an intronic TTTA, which is a reference repeat sequence, usually typically quite small, maybe 5 to 7 copy numbers, and an inserted TTTCA repeat expansion. And these are expand to many hundreds of copies that all are associated with these 6 genes and all cause FAME. So I'm going to tell you a bit about our story and how Bionano has helped us to learn some new things about this disorder. And a lot of our story is linked to this large Australian, New Zealand pedigree collected by our collaborators Ingrid, Doug and Lynette in Melbourne and New Zealand. And a lot of the linkage work which I'm presenting on this slide was performed by Melanie Barlow's group. So here we started out our story in around 2012, and obviously collection of this large family started well before then. And we can see in this original work that the linkage was narrowed down to 13.3 megabase region, spanning the centromere on chromosome 2. We formed an international collaboration with individuals in Europe and the US, and obviously the Australian groups. And we formed an identity by descent mapping. So we looked at whether haplotypes were shared across multiple FAME families. There are about 20 odd different families affected by FAME. And we were able to use that to show that there were several founders and that these were and helped us narrow the linkage interval down by another 3 megabases. There's still quite a lot of genes in this region. And then finally, just before this time last year, we were able to use various sequencing techniques to identify the repeat expansion which causes FAME2. And that was quite a relief for many of us. So what I'd like to do is just briefly show you some data from that paper. So this is what the FAME repeat expansion looks like on short read whole genome sequencing data. So this is probably what most people would use to look at this kind of situation where you have a large unresolved family. And you can see this is an IGV shot. So we have everything that's grey matches to the reference, and then these reads which are in green and red, or green, orange and red, are indicating reads which are not mapping to the reference, and that's because they contain this repeated sequence. You can use short read sequencing techniques to measure the sizes of these, but there's some problems with that method in that it doesn't quite get the right size. So I asked the question for the course purposes of this talk, what if we had used Bionano instead of short read sequencing and whole genome sequencing in 2016? And so in collaboration with Jill Lay and Alex Hasty at Bionano, we're very thankful for their cooperation with this work. We performed Bionano assemblies on 4 individuals from this large Australian New Zealand family. And here you can see the typical mapping data. And of course, this is for chromosome 2. And I've just marked the unaffected allele in this darker blue and the affected allele in this lighter blue, and the green is the reference map from Bionano. So on de novo assembly, we see that every one of these individuals has a repeat expansion on chromosome 2. And in fact, if we overlap them all across in something like the UCSC genome browser, we can see they map directly into the first intron of STAR-D7 and directly where this AAT repeat expansion occurs endogenously in the genome. So basically, in one experiment, we would have found exactly where this mutation was, gone from a 10.3 megabase linkage interval right down to a couple of kb, and found this expansion very rapidly. In the last few minutes, I'd just like to quickly go through some of the data that we're moving forward with on this project. So we have expansions, now a method of measuring the repeat expansion across multiple individuals using long-range PCR. And here we can see successive generations of this Australian family that we do see increases in size of this repeat through the germline. and that this is correlated with work that we published showing that there is clinical anticipation in this family where disease onset is becoming earlier in those same 3 generations. We've looked also at the inheritance of the repeat, maternally or paternally, and we don't see any germline specific effect from the parent of origin. In terms of looking across the population more broadly, so multiple individuals affected with FAME, we've measured, it's been measured in FAME 1, and Crystal, I'm sure we'll talk about FAME 3, that the size of the repeat is anti-correlated with the onset of the myoclonic or the epilepsy. So basically, the longer your repeat is, the much earlier the disorder will onset. So just to briefly summarize our FAME 2 story. It's caused by repeat expansions of TTTA, which is reference, and TTTCA, which is non-reference. We've screened that in 158 clinically affected individuals over 22 different families, and they all tested positive for this repeat in STAR D7. We have anticipation, which is observed across all forms of FAME that we've looked at to date. There is both germline, and I'm sure Crystal will talk about the somatic repeat instability in FAME. And the repeat length and copy number is anti-correlated with the ages of symptom onset, particularly the TTTCA repeat content. And of course, Bionano optical mapping is a very efficient way to identify novel insertions, and certainly a technology that you should consider in some of these unresolved families. And then I'll just quickly flash up our slide, thanking a lot of the people that have been involved in this wonderful project. Thank you very much.
So good evening, everyone. So first I'd like to thank Bionano for the invitation and also for giving the opportunity to present this work on familial adult myoclonic epilepsy type 3. So I'm Christelle de Pienne. I work at the Institute of Human Genetics in Essen, Germany. And I'm really delighted to share this presentation with Marco Beck, who has also been a very nice collaborators for us. So you just heard Mark presenting a nice overview on repeat expansions and repeat expansions causing FAME, but I just want to remind you some important characteristics of these expansions. So familial adult myoclonic epilepsy is a dominant disorder that is characterized by an adult onset myoclonus associated with epilepsy in at least half of the cases, and it is associated with the same repeat expansions in at least 6 different genes that you can see on these slides. And these expansions are composed of 2 different motifs, a TTTA motif, which is here in yellow, and which is actually the reference repeat present in normal individuals, repeated between 10 to 20 times. And the expansions also contain a repeat insertion of a different motif, 3TCA, which is depicted here in red. And this motif is probably the pathogenic part of the expansion. And here I'd like to mention that in some healthy individuals, some of the TTTA is expanded, and that it's non-pathogenic. So it's probably the TTTCA that is associated with disease. And so all these expansions are intronic, and all genes are expressed in the brain. But if you look at the encoded products of those genes, they all have different functions and cellular localizations and have really nothing in common. And so it's pretty clear that these expansions are probably pathogenic independently of the gene where they occur. And the gene itself is just a way to express the expansion. So I will talk about FAME3, so expansion in the gene called MARCH6 or March F6 because it has been renamed recently. And the story is very similar to Mark described for FAME2. We first mapped the locus in this very large French family more than a decade ago. And then there was a single locus on chromosome 5, encompassing 9 megabase and about 50 genes that we sequenced many times over the 10 years. And we couldn't find what was the causative variant until the FAME1 repeat expansion was found. So rapidly after, we could identify the expansion in March 6, confirmed it using repeat prime PCR assays, as you can see on here, and find additional families where this repeat expansion occurred and was associated with FAME. And of course, the first thing we wanted to learn about these repeat expansions is how large they were and what was also their structure. And then we used 2 different techniques. The first one is nanopore sequencing, and I will present the results. So if you're interested in those results, I would refer you to our paper published last year. And I will take a few minutes to present another method that we use to characterize these expansions, which is molecular combing, also known as fiber fish. And this technique is actually quite similar, so it has a common characteristic with Bionano optical mapping. So the first one is that you cannot use the DNA that has been stored in your freezer for many years. You have to have fresh blood samples or living cells to perform new extractions. And what you do is to extract very long fibers and you comb them on cover slips, and then you simply do FISH, fluorescent in situ hybridization, and we very simply labeled the region where the expansion occurs in blue and green, so the flanking regions, and we also labeled the TTTCA part, pathogenic part of the expansion, in red. And this is the result in healthy individuals, who you see always the blue and green stainings, and you never see red. And this is what you see in affected individuals with the expansions. So you see the same flanking regions with this red region in between, and you can appreciate the variability between different individuals which are here, and it's a quantitative technique, so we could use it to really quantify each part of the expansion. And looking at this individual who had the largest expansion with the problem of one of the family, we could actually see an amazing variability of the repeat expansions that you can see on these pictures. So some repeat expansions have no TTCA, some are interrupted, some are very large, and there is really an amazing variability that you can see also here under measurements. We also described actually some abnormalities, as we call micro rearrangements, where the red part was not between the blue and the green, or signals that had overlays. And we used these measurements to do the correlations between the age at seizures or tremor onset, and to show that actually it was the TTTCA part that correlated with the onset of epilepsy in our family. So what we did is, so just to come back at this slide, so this is a very nice technique, but the drawback of this technique is actually that it's a lot of work to develop it just for one assay, one regions, and we have at least 6 different to study in FAME. And so, of course, we were interested in Bionano optical mapping because of its ability to detect all FAME repeat expansions at the same time, the genome-wide level. So first, we wanted to be sure that the technique is able to detect those expansions. So we took the same agarose plugs that we use for molecular combing, and of individuals of different sizes of expansion. And so we sent the samples to San Diego, and they did the experiments. And they extracted the DNA from agarose plugs. They labeled it with the DILI1 enzyme, and they performed an assembly and also a de novo assembly and a rare variant analysis pipeline. And of course, the questions were, is optical mapping able to detect the repeat expansions that we saw in these patients, and do the results match the previous findings that we obtained with molecular combing? So for those who don't know, we've never seen a result from Bionano, just to mention that actually this is a very powerful technique that is able to identify really many CNPs, and so you should be prepared, you know, to filter structural variants. And you see, if you don't filter all the structural variants, that you can see more than 4,000 variants of different types. So what we use is actually the control individuals that Bionano offer. So you can use 200 control individuals to filter your data. And if you do this, and also you keep only the structural variants overlapping genes, then you find a number of structural variants that you have to look at, which is really decreased, or let's say about 30 CNVs or structural variants altogether. And here it is, the insertion that corresponds to the repeat expansion, and it's actually the only structural variance that is detected in our locus, and also it would be the only structural variant detected in more than one individual. And this is how the expansion is seen by optical mapping. So it's actually the same individual for which you have seen data previously, the one with the very large expansion. So using Bionano, we could detect an expansion of 40 kb, which was exactly the same size that was detected by molecular combing. And you can see when the molecules are sorted according to their size that actually they are all different. So the somatic mosaicism is really really huge, as we previously we saw with molecular combing. And I want to just highlight here that there are several molecules that are broken just at the site of this expansion, and we think that it's the repeat expansion actually constitutes a fragile site. So I said that we also use the rare variant analysis pipeline. This is what you see on this part of the slides. And as you can see, this pipeline is also able to detect the somatic mosaicism, although the molecules are beamed into arbitrary categories, as you can see here. So what about the other samples? So the good news is that we detected the expansion in all samples. So for those samples, we had a bit of variability of the mean size of expansion compared to what we detected or what we calculated with molecular combing. So here with Bionano, we had 5 kb in this individual for a size of 7 kb previously calculated. We could see also the somatic mosaicism in this individual, and this 13 kb using Bionano, 6 using molecular combing, and we also detect the somatic mosaicism. And then in this individual who had the smallest repeat expansion, calculated around 5 kb, it was actually 2.7 kb detected by Bionano, and no mosaicism was detected because the rare variant analysis pipeline detects mosaicism for structural variants above 5 kb. So my conclusion, optical mapping is a very efficient technique that can detect the same repeat expansions at a genome-wide scale, which is extremely useful for us because we don't have to develop different assays to diagnose these repeat expansions. We could confirm the somatic mosaicism using both the rare variant analysis pipeline and also just looking at the molecules themselves. If you want to do gene discovery with Bionano, you really need control individuals to filter the variants or to just analyze multiple samples or have an idea what you are looking for, because you detect a lot of structural variants with the techniques. And the drawback of Bionano is, of course, that you cannot detect what is inserted. And in the case of FAME, it's important because we really need to know that there is a TTTCA repeat expansion part that is the pathogenic one. So we need to combine optical mapping to different technology, a sequencing technology, to be really able to say that there is a TTTCA repeat expansion at this site. And finally, I just want to mention that the resolution of the technique is around 500 base pairs, which is sufficient to detect the large repeat expansions, but which is actually insufficient to detect the small repeat expansion, including all or most of the polyQ, polyglutamine, polyalanine expansions. And this is an overview of the repeat expansion disorders. And just to highlight the ones that could be detected by the technique, it's the ones that are in green. So these ones would be for sure detected because they are above the threshold for detection. The orange ones are the ones that could be detected if they are large enough, but some of them could be missed because they would be too small, and the red ones would not be detected. And in the case of FAME and also Canvas, which is another expansion, repeat expansion disorder, there is the problem that the pathogenic motif is actually different from the reference motif, and some individuals have non-pathogenic expansion. So, of course, it's important to make the difference. And I will finish my talk by thanking all the people who were involved in this study over so many years. So people from my group, Raël Florian, Sabine Kaya, Cécile Saint-Martin, Caroline Nava, Boris Keren, Christopher Schroeder, and the FAME consortium, including Mark and Joseph, Melanie, Ingrid, and so many others that are not even on these slides. Big thanks to Family and to funding agency, and also to Bionano for helping us with these results. Thank you, and happy to answer your questions.
Good morning. I would like to thank the organizer for inviting me to report our experience with optical genome mapping on cases presenting with repeat expansion disorder and complex chromosomal rearrangements. We have been starting with this technique for a few months. And first of all, we decided to run 30 known samples just to see whether and how this platform could actually detect known imbalances. So we selected samples accordingly with their chromosomal or clinical indication, and we selected 5 samples harboring an unbalanced chromosomal rearrangement, 7 with a balanced chromosomal rearrangement, 7 cases with copy number variants, such as deletions or duplication ranging from 44 kilobases to 600 kilobases, 3 cases with nucleotide expansion, and in 8 of our cases we were interested in knowing whether in candidate gene there was some kind of structural rearrangement or small deletions, because these samples, these patients had a very clear and strong clinical indication suggesting a specific gene condition without a molecular characterization. During this presentation, I will focus my attention on 4 cases. The first one is a case with an unbalanced translocation, reciprocal translocation, and the 3 cases we have with nucleotide expansion. The first patient is a female, 6 years old, with a drug-resistant epilepsy and psychomotor retardation. Cytogenetic analysis disclosed a reciprocal translocation among the long arms of chromosome 2 and 18. Here on the right you can see a FISH analysis with painting probes, green for chromosome 2 and red for chromosome 18, so you can see the translocation. We also tried the samples, processed the samples with array CGH, which disclosed a small deletion on the long arm of a chromosome 2. The deletion arose de novo, such as the translocation itself, so we thought that it could be the breakpoint of the translocation, even because it was exactly where we thought it should be the breakpoint for chromosome 2. It was 181 kilobases inside, and it involving one known gene, a sodium channel gene, SCN2A. This gene is disease-causing gene, and it is associated with epilepsy and psychomotor retardation. The strange thing is that normally gain-of-function variants in this gene lead to a severe early onset phenotype. Normally, loss of function or deletion, as in our case, result in a later onset phenotype and a milder phenotype. So it was strange that our patient, which was 6 years old, presented such a severe phenotype in association with the deletion of this gene. But as we know that this kind of phenotype is very variable, we thought that maybe we were in an extreme side of a broad range of phenotype. And we thought that this gene could be causative for our patient phenotype. We selected this patient for analyze it on optical mapping because we wanted to see whether the platform could be able to detect both the translocation and the deletion. And what we saw is that the rearrangement was actually more complex than expected. In fact, as you can see here, the circle plot, we have an interchromosomal rearrangement with one breakpoint on chromosome 18, as expected for a reciprocal translocation, and two different breakpoints on the long arm of a chromosome 2. The region which is involved between the two breakpoints is inverted on itself. We can visualize these rearrangements in detail just by clicking on them. So here I'm clicking on the inversion. I can see immediately the breakpoints of my inversion, and I can visualize an enlargement of the region. So as you can see here, we have the inversion. The green segments are the references for chromosome 2, and our patient's molecules are represented as a blue segment in the middle. As you can see, this inversion disrupts a different sodium channel gene named SCN1A. This gene is not deleted, it's just disrupted by the inversion, so it's a new information that we didn't have before. What about our deletion? It is detected by this technique as well as this red line in the structural variant portion. And if we click on it, we can see an enlargement of the deletion. We can see that the size is more or less how expected, and that it involves SCN2A gene as already detected by array CGH. We can do the same thing for the other rearrangement. We can see the interchromosomal rearrangement and the two different breakpoints. We can open one by one. We can see the breakpoints and we can visualize the details, and we see that these rearrangements do not involve any known gene. The same thing for the other one. No known gene involved in the translocation. Again, you can see the references in green for chromosome 2 on the top and for chromosome 18 in the bottom. In the middle, we have the molecules for our patient. So what happened to our patient? The more likely is that 4 different breaks occurred on the 2 involved chromosomes. The first one on the long arm of a chromosome 18, and 3 further on the long arm of a chromosome 2. The first break, starting from the centromere, is where the deletion occurs. And 2 more distal breakpoints with the included portion which is inverted on itself, and the terminal portion of the chromosome which translocates with chromosome 18. So this is how it appears, our derivative chromosomes. And the most important thing is that here in the breakpoint of the inversion, we have the disruption of SCN1A gene. This gene is a disease-causing gene, a known disease-causing gene, which is associated with Dravet syndrome. This is an autosomal dominant condition, which is associated with epilepsy and psychomotor retardation with an early onset of seizures. So this gene fits very well with our patient's phenotype, and maybe is this one the real cause of our patient's phenotype, and not the deletion of the other sodium channel gene, which only partially could explain the severe phenotype presented by our child. The second patient that I would like to present is a patient presenting with a Friedreich ataxia. Friedreich ataxia is an autosomal recessive condition, normally caused by an homozygous repeat expansion in FXN gene on the long arm of a chromosome 9. The onset is normally during the puberty, and the first clinical sign is normally an hypertrophic cardiomyopathy. It is also associated with a severe ataxia, which is progressive with the age, and other signs such as optic atrophy or scoliosis. Our patient is a female, 9 years old. She only presented with hypertrophic cardiomyopathy at that age. We already knew that she had a Friedreich ataxia because a specific assay was performed detecting homozygous pathogenic expansion in FXN gene. It was 2.5 kilobases in size. When we processed this sample with optical mapping, we saw that our patient also presented a large deletion on the long arm of a chromosome 9, which is inherited from her healthy father. The deletion involved several genes, including, of course, FXN. So one allele of this gene was completely deleted. And the software also detected the expansion as a blue bar. You can see it here. If we click on it, we can have the detail of this aberration. You can see that it involves the first portion of FXN gene. It is called by the software as an insertion. That's because the software sees that the two neighboring labels here on the reference chromosome are a little bit more spaced on our patient. So it detected that something is inserted there, and it's actually our nucleotide expansion. So optical mapping allowed us to determine that in this patient, the phenotype was explained by an hemizygous nucleotide expansion because one allele was completely deleted. The deletion was inherited from the father, and the other allele presented with the nucleotide expansion. Our second patient is a female, 15 years old. She also presented with an hypertrophic cardiomyopathy, but because of the age, she also presented with ataxia and scoliosis. She was no more able to walk. Again, a specific assay for Friedreich ataxia detected an homozygous pathogenic expansion in FXN gene, and it was confirmed by optical mapping. So you can see here the insertion involving the first portion of FXN gene. It is in homozygous status, so we can see both the causative events. The size, again, is 2.5 kilobases. The third patient is a patient presenting with a Fragile X syndrome. It is X-linked syndrome caused by an expansion in FMR1 gene. This patient was already tested by a specific assay confirming the clinical indication of Fragile X, and we wanted to verify whether such a small insertion could be detectable by this platform as well. And it was. You can see here we have FMR1 gene, and the first portion of this gene is involved in an insertion event because the labels here on the reference are a little bit more spaced in our patient. It is reported as an homozygous expansion. Actually, it is in hemizygous status because we are on the X chromosome of a male patient. But the aberration could be confirmed by this technique. So in conclusion, we are very satisfied of the results we had by this technique, and we started to introduce this technique in our routine diagnosis. First of all, for characterized genetic aberrations, because as seen for the first patient I presented, it really can make differences in cases of complex rearrangements where we want to identify the real molecular driver of a specific clinical condition. We are also using this technique in case of recessive pathologies, when, for example, we found a sequence variant in one gene that could explain a phenotype, but the pathology is recessive, so we need to identify the second hit. And we are using this technology to identify small deletions within the candidate gene or structural variants, which could lead to the expression of the phenotype. The same thing for autosomal dominant condition, when we have patients with a very strong clinical indication addressing towards a specific gene, which was not positive for a sequencing analysis. So again, we are interested in that analysis to see whether this gene harbors some kind of deletion or a structural rearrangement explaining the clinical phenotype of our patient. At last, we started a new project on children solid tumor, mostly brain tumors. So we have biopsies of these patients, and we want to run these samples on optical mapping to see whether some rearrangement, complex rearrangement is present, leading to the formation of a causative gene fusion, which could explain the acquired phenotype of these children. Thank you for listening.
Hi, this is Aaron Bossler. I'm a clinical pathologist at the University of Iowa and direct our molecular pathology laboratory. Today, I'm going to be talking to you about our experience with optical mapping for the molecular diagnosis of facioscapulohumeral muscular dystrophy, or FSHD. I don't have any disclosures to present. So the objectives of my talk today are for us to understand the clinical manifestations and etiology of FSHD, outline the molecular testing goals that we have, and then compare and contrast the results from optical mapping with that of traditional Southern blot analysis. So the clinical features of FSHD include that this is an autosomal dominant myopathy characterized by progressive muscle atrophy and weakness. Prevalence is about 1 in 10,000 to 20,000. And the disease manifestations and age of onset are variable depending on the mutation. Symptoms typically arise in adolescence, with over 90% of individuals being affected before the age of 20. There are presentations that are severe infantile FSHD, and some individuals with mutation may remain asymptomatic. So it's more likely to be in females. But for the most part, the disease course is progressive, with 20% of affected individuals requiring a wheelchair. FSHD affects characteristic muscle groups, in particular, facial muscle weakness is one of the predominant signs in individuals who manifest FSHD. One of the other typical finds is with the lower trapezius and the development of upward scapular winging. There are also extramuscular manifestations with retinal vasculopathy, sensorineural hearing loss, chronic pain, and in severe infantile FSHD, usually retinopathy and hearing loss, developmental delay, intellectual disability, seizures and more rapid disease progression. So the cause of FSHD really boils down to the aberrant expression of a gene called DUX4. DUX4 is a homeobox transcription factor that is present on chromosome 4, whose expression is normal in early developmental stages. But beyond that time point, it's very toxic to the cells that it's expressed in particular muscle cells, but other cells in the adolescent and adult individual. Now, it's taken us a long time to get to this point of understanding this very brief statement. And part of this is because the DUX4 open reading frame overlaps with a repeat array referred to as the D4Z4. This repeat is about 3,300 base pairs in length, and normal alleles have anywhere from 11 to 100 repeats that are usually hypermethylated, which suppresses the expression of the DUX4. But deletion of these repeats, or contraction of the repeat array, to 10 units or less leads to relaxation of the chromosomal structure. And with this relaxation, you get derepression of DUX4 and transcription of the open reading frame from the last repeat element. Additionally, you have to have a functional polyadenylation sequence in the last open reading frame of the gene. And sequencing has identified that there's two primary haplotypes. The A haplotype contains the functional polyadenylation sequence following the DUX4 exon. And so you get translation from the stable mRNA transcript. Whereas the B haplotype sequences have variations in the signal sequence, or it's completely absent. And with this loss, you don't get functional DUX4 transcripts. They're degraded prematurely. And so there's no FSH, no protein expression, and these are considered non-permissive alleles. These sequences are different enough that you can use southern blot probes to differentiate these two primary haplotypes. Another complicating factor of molecular diagnosis is that the D4Z4 repeat has a homologous sequence on chromosome 10. These share 98 to 99% sequence homology. Now, they can be distinguished using restriction enzyme digest and southern blot analysis because there are different restriction sites in the 4-type alleles from the 10-type alleles. There's an XAP restriction enzyme site in the 4-type alleles and BLN restriction sites in the 10-type alleles. So that when you perform southern blot analysis and you hybridize to this gray area here that's referred to as the P13E11 probe sequence, when you digest with E, which we found in our validation to be true. So the process here is that you have to isolate high molecular weight DNA. We've done this using our own internal method and by the specimen processing kit that Bionano has provided to us. And then you can label the DNA, fluorescently tag it, either using a nick label system or a direct label system. The nick label system actually cuts one of the strands of DNA, and in so doing, potentially fragments the DNA further. So we opted for the direct label method, which doesn't cut the DNA and just directly labels, puts the fluorescent tag on the DNA. Once the labeling is completed, then we load the DNA onto the chip or the cartridge, which is full of nanochannel arrays, so that we can separate the individual molecules and then perform imaging to take pictures of the fluorescently tagged DNA. Once the separation is completed, the algorithms convert the images into molecules, and then these are aligned de novo, kind of like a restriction fragment length pattern would be. And then these can be compared against a reference sequence, the green bar here, to identify here the sum of all of your optically mapped molecules to see, in this instance, where the labels are separated, indicating that there's an insertion. Here's an example of some of these molecule maps. The molecules are indicated in the amber and the blue marks are where the fluorescent tags occurred. The summative sequence is shown here in blue, or the labeled map, compared to the expected labeling of the reference map here in green. When we look more closely at the end of chromosome 4, where the D4Z4 repeat array is present, using the direct labeling method, there are fluorescent tags at the beginning and the end of the array, but none that occur within the repeat array. And so you actually have to measure the length of the array, subtract out the non-repeat sequence, and divide by 3,300 to get the number of repeats that are present. And that works pretty well. And I'll show you our data here in a second. The other ability, though, is that the permissive A haplotype and non-permissive B haplotype sequences are pretty well discriminated by the fluorescent tags, as shown here in the red and the green boxes. And so in one labeling, you're able to figure out the number of repeats and the haplotype sequence. Now, Bionano has also built a specific analysis pipeline to look at the repeat sequences present on chromosome 4 and chromosome 10. And they also built into this the analysis of the SMCHD1 locus on chromosome 18. There are reported deletions and rearrangements of chromosome 18 that have contributed to mutations in SMCHD1. And so being able to tie this into a single analysis is very helpful. And so the program calculates and reports the number of repeats, the chromosome that they're present on, and the haplotype sequence. And then the chromosome 18 status is shown separately. It takes about 2 to 4 hours to do this computation once the separation has been performed, in comparison to looking at the entire genome, which can take 16 to 24 hours. So when we look at our validation data, looking at the sizing first, we found that the optical mapping had very high correlation with the southern blot analysis. The graph here shows the sizing by the Bionano on the x-axis and southern blot analysis on the y. And we had 146 unique alleles representing from 1 to 79 repeats that showed very high correlation, greater than 94%. That you can see here with the smaller alleles, the sizing is much tighter versus the larger alleles. And in fact, alleles greater than 145 or 190,000 base pairs by southern blot were not able to size by southern. And so we couldn't do an adequate comparison of these. And there were 17 of those alleles. And these are both chromosome 4 and chromosome 10 alleles. Furthermore, when we look at the A and B haplotyping, we had 163 total alleles, again, chromosome 4 and chromosome 10. And we had only one of these alleles not be able to discriminate. And that was more because of the poor quality southern blot and our inability to go back and repeat any of the testing because there was no sample left. So very high correlation with what we expected for haplotyping based on the southern blot result. Furthermore, the optical mapping allowed us to discriminate or find alleles that we weren't necessarily expecting to detect. And these fell into 2 categories. There were 5 total alleles. One of these, the optical mapping showed us that there was actually a low-level mosaic allele, a 5th allele, that was just too low of a signal to pick up by southern blot. The other 4 cases, though, had alleles where the sizing, and that's what's shown in the example on the right-hand side here, for this chromosome 10, these 2 alleles are very closely matched in size, and we weren't able to discriminate this on the southern blot. It looks like a single band. And so there were 4 instances where we were able to identify what appeared to be one allele by southern blot as 2 different alleles because of the optical mapping patterns. And then we had one example of a chromosome 18 structural abnormality. This is the Circos plot that you see with the de novo analysis, identifying this deletion here on chromosome 18, which included the SMCHD1 locus. It was something that we expected in this case, but it clearly demonstrates that deletion. So when we look at those 5 different goals of analysis, the optical mapping performed efficiently and effectively for 3 out of those 5. It can measure the number of repeats, it can determine the permissive 4q A and B sequence status, and it can discriminate the presence of chromosome, the repeats on chromosome 4 or chromosome 10. It's not able to discriminate the 4 and 10 repeats themselves, and we're not currently able to do the methylation analysis. And so then lastly, when we look at the impact on the laboratory from a workflow and a management standpoint, we've determined that the optical mapping is about half the cost of Southern blot analysis, that there's a lot less tech time involved in doing the optical mapping. The specimen requirements are significantly lower with the amount of blood that we have to use, and our turnaround time is much faster in doing the optical mapping. We're also trying to get rid of radioactivity. So this has significantly reduced our use of radioactivity, and it's improved our overall quality assurance because the size discrimination is so much better with the optical mapping. So in summary, optical mapping showed strong correlation with Southern blot analysis with the D4Z4 allele sizing. There's near perfect correlation with the Southern blot for the haplotyping. It resolved the ambiguity for cases with multiple 4-type alleles detected by Southern blot. It allows us to detect alleles that are not detected by Southern blot, so there's a higher sensitivity for low-level mosaic alleles, and can discriminate down to ±1 repeat. We can also detect the structural variants for the SMCHD1 locus, and in our reproducibility, there's very high precision and repeatability in our repeat testing. So I'll just wrap up by thanking the members of our laboratory who were involved in this validation. Aaron Stentz was the primary driver of all the test development, and our molecular fellow, John Thomason, was closely involved in the data analysis. John Priestner, our molecular lab advisor, supervisor, who was integral in getting the instrumentation into the laboratory, and Dr. Steve Moore, who oversees the sign out of all the clinical diagnostic testing that we do. Thank you.
Hello, everybody. My name is Jeroen de Preeuw. I work at the University Hospital of Leuven in Belgium. I'm working at the neurological diagnostics lab. So we're focusing on different disorders, such as FSHD. And during this presentation, I will talk very briefly about the Bionano optical mapping that we're currently using, and also some comparison with the Southern blot analysis. It was asked to just give a small presentation, but for the, I would also like to refer to the presentation of Barbara de Waele. So she's presenting, she presented on the 12th of January about leukemia, and she already did quite some validation about the instrument. We are currently working now on FSHD validation specifically, and so so we are only having yet the first 2 chips loaded, and we're hoping to have later on having more data, so we can present a bit of more data. We can actually also have the entire FSHD validation finished quite soon. And we're also planning to be at the EMQN validation. So it's actually an organization from Europe that sends samples to different labs, and so we can actually compare our results with other labs that are also doing FSHD detection. So now we're actually here. So we already ran 2 ships, and we're hoping in April this year that we are actually finished with the entire validation. So unfortunately, I can only show about 5 samples because we did 1 repeat between the first and the second ship, and so most of the samples we have the data from the southern blot already available. I also would like to mention that we work in collaboration with the University of Leiden. So for us, when we have a result that's difficult to interpret, or we are not quite sure, we send our samples also to Leiden so we can have a second opinion. So therefore, some samples will actually have multiple data available, but generally speaking, the results are quite the same between our southern blot and the samples from Leiden. So we have 2 samples that were affected with 8 repeats, 1 normal sample, and 2 samples that were actually in a gray zone that for us it was not sure where exactly the number of repeats were, but this actually could be detected in more detail if we would like to. So for the first person with the 8 repeats, we have a very straightforward report from the Bionano, and also here we see that the number of repeats was actually 8 in 4QA. So here this person was affected. Also there was a number, there were 23 repeats in 4QB and 21 repeats in 10QA. And here also in this report you can see the coverage of, for example, the 4QA region, and then you can clearly see which DNA fragments are aligned to the region. Also here for 4QB you can see the results. And also very interesting for us is that you can see whether there are some copy number variants in the SMCHD1 gene. So this could be relevant if you would like, if there would be a deletion, then this might be, this patient might have FSHD2. So here you have the southern blot results, and we have from this, we do have the index sample, and we know that this patient has 27 kb, and here you can see here a band, and here it's not that clear in the picture, but if you see it in real time, it would be the results were much better, and also the sample FSA, this patient also had the same number, the same length of bonds. So this is actually in concordance with the Bionano results. We did 2 different chips with this sample, and we find exactly the same number of repeats, both in 4QA, 4QB, and 10QA. Then also the second and the third, yeah, so this patient was concordant. Then also the second and the third patient, we were concordant. However, there was a difference of 1 repeat, but it's actually the sensitivity of the southern blot is ±1, so it's possible that this is actually there somehow a different number of repeats. So, and then the normal sample, we saw that actually 33 and 46 number of D4 sets for repeats in the 4QA of the 4QA haplotype. Then the 4th sample, there we observed that it was actually at the number of repeats was 13, 4QA and then 51 also at 4QA. So it was, for us, it was very clear with Bionano that actually this patient is normal and doesn't have a reduced size. Then one interesting, the last patient was very interesting because here it was actually found that the EcoR1 fragment was 41 kb. So normally the cutoff is at 40 kb, and so this might be caused, the longer 1 kb might be caused by a 4QA long allele. So these patients have a mild phenotype. As you can appreciate from this figure, so this is the 4QA haplotype, and then here you have a long haplotype, and there it's actually a bit longer compared to the 4QA allele. But when we ran a Bionano, we saw that the haplotype was actually 4QB. So for this patient, there was a reduction. So it was indeed 10, but it was 4QB. So here, the patient was actually normal. So that's very interesting because otherwise it would be quite complex. But now we know for sure that it's, or at least I think we need to further investigate this. But it looks like the patient definitely is not affected of this 4QB because it has the 4QB haplotype. So these two patients where we were not quite sure, we observed very straightforward with Bionano that the two patients were actually normal. So as a conclusion, so the Southern blot results are highly concordant with the Bionano results. Although Bionano has a lot of advantages, so it's very sensitive. It's also very robust because with our Southern blot, it's not always that straightforward. We're working with radioactive probes, and this might be sometimes have less, can be less detectable. Also, we have the 4QA and 4QB alleles, and the CMV detection of CMCHD1, and but that's not ready available. But it would be very interesting if it's possible to detect the methylation profile of the D4Z4 repeats, because that way we can also check for FSHD2, and there was one paper published where they used Bionano with methylation-specific fluorophore. So it could be very interesting if this would be developed in the future. And then for us, we will definitely continue with the validation, and we hope that we will have an up and running Bionano test in April of this year, and we will check into more detail the 4QB versus the 4A long repeat. Then finally, I would like to thank our group, so of the University Hospital, also Barbara de Waele and Wendy Martens for helping us out with the protocol of Bionano, and also the group of Joris Vermeers. And then finally, the group of the University of Leiden for also having the FSHD detection. I would like to thank you all very much for your attention.
Okay, can somebody hear me? Hello? Yes. Okay, welcome everybody to our live Q&A and panel discussion. And so I hope all of you can appreciate our excellent speakers today who really demonstrated and showcased their data on repeat expansion disorders, repeat contraction disorders, and complex constitutional cases. We have all of our speakers except Dr. Mark Corbett live with us right now. And this is simply because in Adelaide, Australia, it is really very, very early in the morning. So, with that, I would like to introduce Dr. Sven Bockland, who is our Vice President of Communications and Scientific Affairs, who will be moderating this Q&A with me. So Sven, should we start with the first technical question for Dr. Bosler that we have?
Absolutely. Yeah, hi, good morning or afternoon. Dr. Bosler, we had an audience question about whether you had to make any changes to the way your samples are collected, how the blood is handled or processed at all when you started using the Sapphire system.
It's a good question. What we found was that the stability of the white blood cells was similar to what we expected with doing the Southern blot analysis. And so we've really not changed our requirement to get us a fresh blood specimen. We have found that at about 6 or 7 days of age, the quality of the DNA that you can obtain does start to degrade to some extent. We were still able to get results though with the optical mapping. So maybe there's a little bit of an extension in time, but we have found that the better quality data gets generated, the more quickly you can process that specimen after it's collected. The one caveat though is that you can now freeze the specimens using the protocol from Bionano and come back and do an extraction sometime later. And that has been significantly advantageous if we have a specimen that shows up right before the weekend or something like that. So that has been helpful. But again, you want to freeze the specimen soon after the collection so that it maintains that viability.
Thank you.
So our next question is for Birgit. So you showed the Bionano data on the carriers for ATX and 10 expansions. So, and you also showed a range of, quite a range in mosaicism. So what did your actual patient's repeat length look like? We've had few live Q&A bandwidth and audio glitches in the past 2-3 days, and I was hoping this would go a little bit better. We had good success yesterday, but it looks like that we might have faced something today also. Could you hear the question, or because we can't hear you? William, is there something that, okay, microphone won't connect. Okay, so apologies. That's we do have some technical issues with Birgit. We'll move to Alex Hoyschen. So Alex, very interesting presentation, great data, but you showed everybody that you just saw one de novo structural variant in your trio analysis, and mentioned that you saw very little false positives. So how would you compare that with other methods is the first question. And then I have a follow-up one too.
All right. Just to check, you can hear me now?
Yes.
Yes, we can.
Yes, excellent. So, I mean, to be honest, we don't really understand the de novo mutation rate for human structure variants extremely well so far. So from all we know from genome sequencing, and mainly from large structure variants that have been previously detected by arrays, CNV microarrays mainly, we know that at least the large structure variants quite rarely happen de novo. So there it's estimated that the de novo mutation rate is probably less than one structure variant per genome per generation. So that explains why in 5 trios, we may only find one de novo event that is larger than 500 base pairs. So in that sense, the Bionano results is exactly in line what I would have expected from the array data and also from some genome data. However, what is really very important is that exactly what I pointed out, that the false positive rate is low. So by definition, false positives that would happen in the genome of the child by using Bionano would look exactly similar to true de novo calls. And we don't see any of those in the example I've shown. So that is extremely good and makes analysis straightforward.
Yeah. And then the next question as a follow-up is that you also kind of like alluded to several other cases that you did not show today. Can you comment on how long are these long repeats in your experience that you have seen, and how these SVs have, or de novo SVs have escaped all the other methods?
Right. So just for the repeats, I mean, we are very much at the beginning, and I'm certainly different from many other speakers in today's session. I'm really not an expert on repeat expansions, but I can tell you so much that our diagnostic experts for repeat expansions are really quite thrilled about the very preliminary data that we have so far. So, for example, for a simple DMPK1 repeat expansions, usually our diagnostic test only states that a repeat is beyond 150 copies. And now for the first time, we get a very good estimate of the actual length of the repeat, and that could actually have clinical relevance, and parts of that are unknown. So for that reason, it seems that already just in the very first pilot data, the Bionano results look so promising that we are about to launch a clinical utility study together with our diagnostic colleagues. So that's what I can tell for the repeat expansions so far. And there is, in essence, no upper limit, while many other, at least the genomic technologies, really have an upper limit to what extent they can still find very large repeats. And there, I think, is quite something unique in the Bionano data. For the de novo events, like the example I've shown from our collaboration with Roland Kuiper, I mean, there it's really the context of the aberration that does not allow short read sequencing, like Illumina, to go over the event. So seemingly, the event or the boundaries of that element are so GC rich or so repeat rich that the PCR elements in Illumina sequencing or in the library construction or in the bridge PCR that is used for sequencing really prevents that allele from being present in the sequencing library or in the final result. While the other example that I've shown for the 17q region, that obviously is one of the most complicated regions of our genome, there I believe all the other technologies so far simply have severe issues with mapping. So there, the extremely long reads certainly help. And I think there's still some time needed to really move sequencing assays to such a level of perfection that we can also find those events, or probably we may continue for quite a while to use optical mapping together with NGS to really understand those very difficult regions of the genome to a greater extent.
Thank you. Thank you so much, Alex. Sven?
Yeah, Crystal, let's try to go to you. You obviously showed your results on FAME, and then you showed some other repeat expansions that you think could be tested with Bionano, but those are the known repeat expansions. How many do you think there are that we still have to discover? And do you think a genome-wide technology like ours could help with that?
Can you hear me?
Yeah, we can hear you.
Okay, so hello everyone. Yeah, so I don't have the answer to this question. I wish I would actually. My own belief is that there are many repeat expansions that remain to be found. The exact number, I don't know, because actually if you look, I think there are many repeat expansions in normal genome as well. So if you look at long read sequencing data, what you find is actually a lot of repeats everywhere. So I think there are many repeat expansions that are probably pathogenic, but that there are also probably a lot that are non-pathogenic, and making the difference between the two is actually very complicated. So maybe Bionano could help also by analyzing control individuals.
Thank you. And then for FAME specifically, I mean, you and Mark both touched on that, but you have these very different genes that do very different things in different parts of the cell, yet similar repeat expansions cause similar disease. So do you think this is a case where the RNA itself is toxic, or what do you think actually is the mechanism of the disease?
So we don't know yet the mechanism. So there are some data from the Japanese group who have described FAME1 on chromosome 8 in the gene called SAMD12. And they showed, because they had brain samples, so they showed that the repeat expansion lead to an aberration in the transcription of the gene and an accumulation of the repeat, and also maybe to RNA foci in these brains. But although the STARD7 and the MARCH-F6 genes are expressed in other cells, we don't see these RNA foci. So I think the question is still open, but it's probable that these repeat expansion lead to toxic RNA, or maybe also to toxic peptides through RNA translation.
Thank you.
Our next question is for Viola. Viola, thank you so much for your amazing presentation, where I think one of the interesting cases was where you showed the deletion and a repeat expansion occurring within the same gene. And you also mentioned that there are several assays that you're validating on Sapphire, which you are thinking of implementing in the routine diagnostics in the clinic. Could you briefly speak and let the audience know about your process?
Hi, everyone.
Hi.
So our future projects with these techniques you're asking me. We have an interesting project about solid tumor in children. We are collecting tumor from biopsy, and we are very interested in running them because we are already processed them with NGS or RACGH, but we still lack some information about structural events leading to gene fusion, which are one of the most important causes for some tumors. So we want to know whether some new molecular driver is present in these children. It's a very interesting field because there's not a lot of data in scientific literature about this problem, this issue. So we are starting nowadays. We still do not have enough data about it. And we are still working on specific pathologies, some such as hemophilia, for example, because about 50% of samples do not have a mutation. They present an inversion involving one specific intron, and it's not so easy to diagnostic it because you have to have a specific homemade primer, and you can only detect the specific unknown inversion, but it's possible that other different structural variants are there. There are a percentage of children which have hemophilia, but do not present a specific genetic driver, so we are collecting also this type of samples.
Thank you. So as a follow-up, right now, I think our understanding is that you are using optical genome mapping as a reflex test. But based on all the data that you showed, in your opinion, how many patients would you use for a reflex first before you move this to be like the first line test?
We are already moving on in using this technology in our diagnostic field because we got confident with this technique using known samples, and we want to start with unknown samples. That's why we are collecting these new kind of samples. We cannot replace all our CGH analysis with these technologies because we process a very huge amount of samples by RACGH, but we are selecting specific cases without a diagnostic to see if this technology can help us in find a reason for our children phenotype.
Audio working this time? No. Okay, we will.
We'll come back. Yeah, let's go next step.
In a few minutes.
Sven, go.
Yeah, we have maybe another question for Dr. Basler, Alka.
Yep. So, Aaron, one of the questions that we have is that you talked about all the different contractions and the permissive and the non-permissive alleles, and then the structural variants such as deletions on chromosome 18 for SMCHD1. How about any translocation involving chromosome 4 and 10? Have you had any experience with those kind of rearrangements and their prevalence in FSHD1 is the question.
Good question. So in the clinical testing that we've done thus far, haven't come across those rearrangements. We've been live with the optical mapping since June, approximately, and we'd probably need to go back and reevaluate the data looking at the de novo analysis for those rearrangements. We haven't done that yet. I'm trying to think of what our percent positivity has been with the typical FSHD1, FSHD2 types of cases. And I think our positivity is around 50% or so detecting those. But no, we'll have to go back and look at the cases that we didn't identify a clear mutation and go back and look at the de novo results.
Thank you. And as a follow-up, are you considering other southern blot assays in your laboratory and validate them on Sapphire?
Oh, definitely. No, it was helpful to hear some of these presentations. We do testing for trinucleotide repeat disorders, and most of our testing is PCR based, but we've always had southern blot analysis for potential large expansions. So our preliminary look at that is the data looks good. The challenge, though, is that most of our archive specimens, it's nucleic acid, it's DNA, and so we have to catch the specimens as they come in to get the fresh DNA and prepared. But yeah, no, that's our intent, and hopefully we'll be successful at that.
Absolutely. Thank you.
Yep.
Sven?
Jeroen Depree, my fellow Belgian. Let's see if we can hear you today. Please say yes. No, and we lost him. Okay, we'll try him again later.
So as we have 7 minutes left, what I wanted to do was, because we have the expert panel here, and we've had an amazing 4 days where we started off with constitutional genetic disorders from, our customers from Europe and China and the United States. And we saw both prenatal and postnatal applications using optical genome mapping. And I think that the strength that every speaker and scientist has shown in the past 4 days is the ability to pick up the unique classes of structural variants at the high resolution that conventional methodologies lack as of today. However, globally, the conventional methods are used as the standard of care. Then when we saw a number of talks on hematological malignancies, both leukemias and lymphomas, and then we saw a lot of work on some research-oriented solid tumor work, and then some laboratory-developed test path for solid tumor work like glioblastoma that Dr. Cole showed yesterday. And today we are focusing again back on, the complex genetic disorders, the rare triplet trinucleotide repeat expansions, and then, of course, our contraction disorders. So just looking at this overview, I wanted to get all of the experts, parting thoughts and comments, and what vision do you see and in the future for optical genome mapping. So we'll start with Aaron, since I see you on the left, and then we'll go down the list.
So our observation of the ability to size, in our case, the contractions, but with any of this, that the resolution is so fine detailed, I think there's significant application in clinical diagnostics. I think one of the challenges is going to be having a greater variety of labeling methods that can be used in this, and with the development that I think you'll see expanded use of the technology.
Thank you, Aaron. Viola, if we can hear you or if you can hear us.
So I think it's a wonderful technique that could give us all comprehension of what happened inside the cell. I am a cytogeneticist, and I think that it's very important that cytogenetics also have an upgrade, because it's still at the same point for several years, and we are losing a lot of information. So I think that this genomic technique can really improve the view of the pathologies in their whole identity. I think it can be.
You spoke the words from my heart. I mean, that is the reason why I joined Bionano, because, I'm a trained cytogeneticist too. So we've been waiting for how many decades, right, to have something at a higher resolution where we can demonstrate equivalency with other methods and better performance. So I completely echo what you have just said. So thank you. Crystal?
Technique could, help resolve really region of the genome that could not be studied so far, especially satellites. So I think we've heard a lot about Fshd. We don't know any other disorder that is linked to satellite expansion or contraction, and there are some other satellites in the genome. So yeah, I'm clearly interested in these very complex regions. So not just repeat expansion, but also centromeres, telomeres, all the repeats that cannot be assessed by other sequencing techniques so far.
Thank you. Thank you so much. Alex?
I mean, a lot of that was already said. So I really see quite some potential for optical mapping to replace all of cytogenetics, truly in a diagnostic sense. And from my personal perspective, just also due to our local system, I think the first step will be set in the leukemia field, simply because current testing requires so many parallel tests. The part we can gain is so huge immediately that would be probably the first arena. And again, the deep coverage that we can gain from Bionano really gives us sensitivity that is unmatched for some of the somatic events. So I think that's, in our view, potentially is the first step. But I see huge potential to stepwise replace all of cytogenetics, including FISH, karyotyping, and arrays. And then for more research studies, I mean, we started with the unsolved cases, but let's just imagine that down the road, Bionano becomes even more trivial. There's a little bit more automation happening. The cost drops to something like, I don't know, $100 to $200 per sample. So then we don't even have to think about those costs anymore. So then it could be very much happening that we run always sequencing and optical mapping in parallel for almost every sample. And the ease of analysis and then to potentially low cost of Bionano would really enable that. So that's my more research-based vision on it. So yeah, I think the time ahead is really exciting.
So I'm really laughing and smiling because you know that I also represent the executive management right here. So that you really spoke what is needed in the field. We certainly will be working on it, and I couldn't agree with you more. So thank you so much. Let's try if we can get some words from Birgit. No luck, or your own?
Yes, hello. This with Bionano, it's very powerful, and it's really great that we are able to use the technique. And a colleague of mine, she's already working for 30 years with southern blot, and she doesn't like it that much, and she's very happy when she will be on retirement that finally there will be a new technique available for us.
Very, very nicely said. I think that the lab techs, we always want our lab supervisors and lab technologists to be happy. So if they get happy about an advancement and, an improvement, then it's success for us. So I want to, once again, thank all of the expert panel speakers, for your time right now, staying till the end for the live Q&A and for excellent presentations. We have had tremendous attendance in the past 4 days, and we'll give you that follow-up via email. But tomorrow is our last day. It is a bonus day. So we have kind of finished our theme for next generation cytogenomics. But tomorrow we are going to have an expert panel discussion on the COVID-19 Host Genome Structural Variant Consortium that is led by Dr. Ravi Kole from Augusta University, but there are collaborators from across the world. And the focus of this is essentially to look at how the host genome structural variants may be playing a role in this pandemic, which is literally affecting every one of us in one form or the other. So for the audience and the viewers, please come tomorrow. And for the expert panels, if you have time, I would really love to see you all over there tomorrow also. And so once again, thank you for your time. And hopefully we'll have more data with all of you speakers next time. Thank you, everyone.
Bye-bye.
Bye.
All right, so that is the end of today's symposium. Of course, going to be holding tomorrow's. It's going to be Friday, 10 a.m. Eastern Standard Time. And before I sign off, sorry, I'm hearing my echo. Before I sign off, I want to do a quick price analysis because they're just soaring up today. So I want to give you an idea of where I think they might go today. So currently, they're extremely bullish, up 25% today, trading at $7. There's a resistance at around $7.14, and currently, based on the volume, the ton of volume that we've seen today, they are forming a wedge with a top right here, both where spikes of volume occurred, and then we have the bottom of this trend line going right here. And I'll actually link this chart for you guys to download, and I'll put that in the live chat. Give me 1 second on that. So if you guys have Thinkorswim, you should be able to download that link right there and see my charts, and that will be updated to you as soon as I change anything on there. Anyway, so my predictions, end of day, it'll probably stay within this wedge if the volume continues to go up as it has been. And if we look at yesterday's volume, the volume is a lot higher today than it was yesterday. It looks to be about 2, maybe even 3 times as much volume. I think this is mostly due to NASDAQ compliance news coming out. If you guys missed that, basically BNGO stayed above $1 for 10 consecutive days, and now they don't run the risk of being delisted, so that was great. But this is also something we should have known a couple days ago going into it, considering they were trading at $5 and falling by 80% was unlikely. So, but I do think that with all the news outlets now posting about this, it's getting a little more people hyped about it, and also with the scientists being pretty optimistic today. I noticed that there was much more optimism from today's scientists than there was yesterday and the day before. So I think that a lot of people are very encouraged by what they saw today and all of the great words that the scientists had to say. So it could continue to go up from here. And by the end of the day, if it stays within this channel, we can track to see where it might go. Maybe as high as around 767. That would break a new high. The previous high was, I think, 714. I might be wrong on that. Let me double check for you guys. So 724 was their previous all-time high. So we might see it break that high today. Toward the downside, if they can't keep this steam going, if the volume maybe dies down for the rest of the day, we could see it trend along this bottom trend line and maybe fall through, possibly getting it sent down back down to around 655. I do think there is a new support somewhere around 677, so that might actually support as well, somewhere around that range. Yeah, they're going to, as Raphael said, they're gonna try to promote the product to its fullest potential, because the thing whenever it comes to these scientists presenting is that they get money from the NIH or wherever they get their sources of income for their research. They get this based on their optimism for the future uses in their labs. So of course they are going to be optimistic about the future of it because they want more money. So you do have to keep that in mind. But again, I just want to say that this group of scientists over yesterday and the day before, they seemed a lot more optimistic than previously. Can this be a $30 stock like PacBio? I don't think that they'll ever reach the valuation of PacBio. Maybe PacBio's current valuation, but I don't think that they'll reach the same valuation just because they have differing technologies. PacBio has multiple technologies, whereas BNGO only has a single one. So you have to keep that in mind. And also, PacB has full genome sequencing, whereas BNGO does genome mapping. So those are completely different, but that's not to say that BNGO couldn't come up with new technologies in the future with all this money that they're likely going to be making, probably through another cash or another stock offering pretty soon. I wouldn't be surprised to see that in the next maybe week or two if their stock price does continue to go up, because they do need some more cash for FDA approval, and a lot of people have been asking me about FDA approval. It's a really lengthy and costly process, so you have to keep that in mind. And Simon from ArcG actually talked about that process in a Twitter thread, and I'll try to find that and I'll link that in the chat as well for you guys. And after this video, after this live session is over, I will link that for y'all. So if we go to Twitter and go to Simon Barnett's page, mentioned FDA, I think right here. So I'll link that, the thread that he talked about FDA regulations, in the chat right now. Stock offerings typically do lower the price to some degree. Generally, it's kind of seen as this way to make money from shareholders, because they're ultimately the ones that end up buying it up. But that's not to say that it couldn't continue up, just like we saw with the stock offering we had the other day. That was about a few days ago, I think. So if we look at the 5-day chart, that would be pretty helpful to see that. So this was when they announced this, I believe. It sold off, but then they held this, and then they went up, and then they kind of traded flat for a few days, and now they're soaring up. So initially, it is going to have a negative effect on the stock price, but if people really believe in the company, if they really believe in the future, then it's going to continue to go up. It's kind of like how Tesla keeps doing their stock offering of $5 billion. Similar in that way. Pretty much any time that Tesla's done a cash offering or a stock offering, they got sent down. I think there was one right here, somewhere in this area. I'm not sure when exactly. but I do know that they went down whenever they offered those stock to the public. What do I think of IsoRay? I'm not sure I've heard of that one. The stream should be good. Maybe try upping the quality in the settings, because I have an excellent connection, so I'm not sure why it might be blurry at all. Yeah, it might take a couple minutes, as MKC said. Anyway, so that's pretty much all I have for you. If you guys want to see the chart that I have for BNGO, I'll show you one last time, and then I'm going to be signing off for the day. I do have some errands to run, so hopefully you guys enjoyed today's live session, and hopefully you're here tomorrow. Tomorrow's going to be the last day for the symposium and the last day of my lives for BNGO. So I really hope to see you there, and that's pretty much all I got for you. So thanks for tuning in, and as always, remember to stay positive, stay green. I'll catch you in the next one. Bye, guys.